Kassady Sheble, and I am a freshman in Occupational Safety and Health.
I'm Kylie Pattison, and I'm a sophomore in Occupational Safety and Health.
I'm Samantha Bacon, and I'm a freshman this year at Montana Tech, and I'm majoring in biology.
And I am Mariah Sheble, and I am a senior in Professional and Technical Communications at Montana Tech.
The misuse and overuse of antibiotics have led to bacteria becoming resistant to antibiotics. How do these bacteria become resistant? Why should we be worried about antibiotic-resistant bacteria? And what can we do to prevent this?
In this video, for the Principles of Living Systems class at Montana Tech, we will be discussing antibiotic-resistant bacteria.
We hypothesize antibiotic-resistant bacteria arise because bacteria who are susceptible to antibiotic will be killed, while those who stand a greater chance against the antibiotic will survive.
We predict if our hypothesis is true, there will be evidence to show bacteria try to become resistant.
According to the Center for Disease Control, or the CDC, bacteria are single-celled organisms that are usually found inside and outside of our bodies. Microbiology Online states the cell structure includes no nucleus or membrane-bound organelles. They contain their genetic information through a single strand of DNA. The bacteria live as single cells, pairs, chains, or clusters.
The human body contains millions of bacteria inside it, while on the outside, the body contains 100 million more bacteria. According to Biology Concepts and Connections textbook, bacteria can reproduce quickly through binary fission.
This is an asexual reproduction produced in which one cell divides into two cells, and then four, and so on. It is used by prokaryotes and some organelles within eukaryotic organisms. In the optimal conditions, bacteria can produce new generations in under 20 minutes. The CDC states many bacteria are beneficial to us. However, there are some that can trigger illnesses, like strep throat or ear infections.
The way the medical field treats these illnesses is antibiotics. The CDC states antibiotics are antimicrobial drugs that fight infections caused by bacteria. Antibiotics refers to a natural compound produced by a fungus or other microorganism that kills disease-causing bacteria. According to the National Academy's Scottish bacteriologist Alexander Fleming discovered the first antibiotic, penicillin, in 1927. The drug was circulated in the 1940s, and the antibiotic resistance arose as early as 1942.
The Alliance for the Prudent Use of Antibiotics, or the APUA, explains antibiotic resistance as an antibiotic losing its ability to effectively control or kill bacterial growth. The bacteria become resistant to the antibiotic and continue to multiply.
Antibiotic-resistant bacteria become resistant through natural selection evolution. They either self-multiply or pass DNA between different bacteria. The CDC says some bacteria have the ability to neutralize or escape the antibiotic effects. That particular bacteria can multiply and replace the others who were killed by the antibiotic.
When multiplying, they pass the genetic code for resistance through DNA coded for resistance. The APUA states bacteria can also become resistant by undergoing a process called conjugation. Conjugation is when bacteria transfer genetic material from one bacterium to another. By passing this DNA, bacteria can become resistant to many different families of antibiotics.
According to Science Direct, bacteria contain error prevention and error correcting systems. One of these is the Methyl-Directed Mismatch Repair System, or MMR system. MMR systems are the location of naturally occurring strong mutators caused by defects in the MMR system. The MMR deficient strains have superior genetic backgrounds for the selection of antibiotic-resistant mutators. If the MMR barrier is removed, the bacteria are more likely to increase the frequency of horizontal gene transfer.
A loss of fitness, as mutators gain deleterious mutations more frequently than non-mutators, is known as a permanent global mutator status. The fitness limitations of mutators can be overcome by high bacterial cell densities achieved during acute infection or the adoption of transient mutators. These mutators are a risk factor during treatment of bacterial infections because they enhance the selection of mutants and have the capacity to refine existing resistant determinants.
Taking antibiotics with just a simple viral infection can cause bacteria to get stronger and stronger. The bacteria changes itself to brush off the effectiveness of the antibiotic. They either can push out the antibiotic as it comes in, balance it out before it has any effect on them, or they can take over the attack site. If bacteria live through the antibiotic attack, then they multiply and produce more and more resilient copies of themselves.
When these antibiotic-resistant bacteria, Science Direct says there are no obvious strategies to eliminate mutations from bacterial populations. We can only try to prevent these bacteria from becoming more resistant. According to CDC, it is important to understand that even though antibiotics are beneficial, they should not be used for viral infections such as cold, cough, or flu.
Resistant bacteria can spread to close members of personal circles. This includes family, friends, colleagues, the community, and others. When it spreads to others due to its new resilience, it can be more harmful and harder to control. It is also dangerous for people who are very susceptible to infection.
Their immune system is already compromised, so if they come in contact with this new strand and get infected, it becomes harder to treat them. Microbes, along with bacteria, can also become resistant. The more drugs someone takes, the more resistant these microbes become, making it harder and harder for doctors to treat infections. This is a concern due to the fact medicine is going to have to become stronger to keep up with the ever-changing resilient bacteria and microbes.
The CDC provides some helpful tips to help prevent antibiotic-resistant bacteria.
Talk with your healthcare provider about antibiotic resistance and ask whether antibiotics are likely to be beneficial for your illness and what else can you do to feel better.
Do not take an antibiotic for a viral infection.
Do not save some of your antibiotic for next time you get sick. Discard any leftover medication once you have completed your prescribed course of treatment.
Take an antibiotic exactly as the healthcare provider tells you. Do not skip doses. Complete this prescribed course of treatment even if you are feeling better. If treatment stops too soon, some bacteria may survive and reinfect.
Do not take antibiotics prescribed for someone else. The antibiotic may not be appropriate for your illness. Taking the wrong medication may delay your correct treatment and allow bacteria to multiply.
If your healthcare provider determines that you do not have a bacterial infection, ask about ways to help relieve your symptoms. Do not pressure your provider to prescribe an antibiotic.
To summarize everything we have said, bacteria are single-celled organisms with no nucleus or membrane-bound organelles. They contain genetic material in a single strand of DNA and reproduce through binary fission. We treat bacteria with antibiotics, which are antimicrobial drugs that fight infections caused by bacteria. Bacteria can become resistant to antibiotics.
Antibiotic resistance is defined as antibiotics losing its ability to effectively control or kill bacteria. Bacteria can also become resistant through natural selection. They can pass on their ability to escape antibiotics through DNA coded for resistance. Bacteria can also go through conjugation or the transfer of genetic material from one bacterium to another to pass on antibiotic resistance.
MMR systems are the part of bacteria where the naturally occurring mutators cause defects. The low fitness limitations of bacteria increase the chance of becoming resistant to antibiotics. We cannot stop bacteria from becoming resistant, but we can try to prevent it from getting worse. Antibiotics should not be taken to cure viral infections such as cold, cough, or flu. This is due to the fact that more and more antibiotics are taken, the more likely the bacteria are to become resistant.
At the beginning of this video, we hypothesized antibiotic-resistant bacteria arise because bacteria that are susceptible to the antibiotic will be killed, while those that stand a greater chance against the antibiotic will survive. After completing research, we conclude our hypothesis was correct. Antibiotic-resistant bacteria arise through natural selection, meaning those who have the ability to survive the antibiotics will pass on that gene to future generations and have a better chance of surviving than bacteria without this type of gene.